here. First question. Oh. Hands up, thanks. All right, so we're either mostly right or mostly wrong. If you answered false, somebody give us the reasoning for why you thought it was false. Alan?
Energy can't be created or destroyed, it only transforms.
All right, we can only transform energy from one form into another. That's also known as the first law of thermodynamics. Good.
No.
I can tell it's you. What, Cameron? There we go. All right. Is this our first?
Unanimous?
Unanimous?
Yes.
All right, good. We're all right. No, even better, we're all right. All right, the reason why an enzyme interacts with its substrate is because it has a complementary shape, but it also has the right chemistry. Good. Right. Ready? All right, so what is it that enzymes do? This is one of the key questions we're going to get at today. We're not adding, right? The enzyme doesn't add energy to the reaction. What it does is reduce the amount of activation necessary for a reaction to take place, right? So we'll look at that in detail today, right? So you can expect to see this question again because I'm going to want to make sure that after we've actually talked about it, we have a better grasp of what that means. So you may have seen some of this before, you may not have, you may have read ahead, right? But we're going to talk about this in detail today. Okay, right, so where did we leave off? We left off with the second law, right?
Yep.
All right.
Yeah, wait.
So, one helpful thing to think about is this thing called entropy. And remember, we defined entropy as disorder. So, if we're increasing entropy, we're increasing the amount of disorder in our system. Right? So one way to think about disorder is the number of possible positions or motions that a molecule could have. So the more movement, right, the more possible places it could be, or it's moving around, that's more disorder. So which is going to have a greater level of entropy? sucrose, or glucose and fructose separate? Hold up one finger if you think it's sucrose, two fingers if you think it's glucose and fructose. All right, not bad, right? It should be this, right? If I have two separate molecules, that's more possible positions and motions taking place than if I just had a single molecule bouncing around. So this is what we would call favorable, and we're going to talk about what we mean by something being favorable or unfavorable here a bit more, right? But just the fact that we increase the amount of entropy means that, this is more likely to happen, to go from this to this. Now, there's other things involved, but in general, we know from the second law, right, that entropy is always increasing. Okay, so what we have here is a very simple diagram of lots of different molecules that the cell makes, right? So here's a bunch of squares stuck together. That's probably a generic representation of a what?
Protein.
Could be a protein. A protein is an example of a
Macromolecule.
but they look like similar units all stuck together. Polymers, okay, good, right. So, maybe this is a polymer, this is a polymer. Right, whatever these macromolecules are that the cell makes, right? The cell has to constantly make these things. Now, what are we building these things from? The building blocks, right? So the monomer units, for example, or whatever chemical building blocks that we're starting. Pathways that involve chemical reactions that build things into larger molecules, right? We start from small, we get to bigger. These are called anabolic pathways, right? And your cells have to constantly do this. They've got to constantly make proteins or DNA, lipids, whatever it is, they're constantly making larger molecules. So, first question, where is there more entropy? Where is there more disorder? Here or here? Yeah, right? There's more entropy here. I have more stuff bouncing around than I have here. So this is actually doing the opposite, right? We're going from more entropy to less entropy. So I have a question. Does this mean that living systems are defying the second law of thermodynamics? No, why not, Alex? Any ideas? That's all right. It's definitely not. So the answer must be that somehow, even though we are reducing entropy here, somehow overall we're making a bigger mess somewhere, right? Right? If we're tidying up, the corner of your room, you shoved everything else under the bed. Right? There's a bigger mess somewhere else. Right? Well, we got to make all this stuff. And in order to do that, we have to get these. But where did we get the building blocks from? Well, we ate a bunch of food. And what did we do with that food? We broke it down through catabolic reactions from these bigger things into these smaller things. All right, so what happened to entropy here?
Increase.
Increase or decrease?
Increase.
Increase, right? And in fact, we must have made a far bigger mess, if you will, right? We might have made much more disorder by breaking down this food than we did by making these molecules from those monomer units. Right? So even though we are decreasing entropy a little bit here, we made a whole lot of entropy just from the food that we took in. Right? And in fact, some of that energy that's there is actually used to power this process. Right? Breaking stuff down generally is going to release energy. Building stuff is generally going to take energy. So, where are we going to get the energy to do this? Well, we liberate it from these guys as we break things down. So we're actually going to use the energy from these molecules to build the other molecules that our cell needs. Right? don't forget, some portion of that energy is going to be lost in the form of heat, right? It's no longer useful energy. So we've increased entropy in general because we went from big macromolecules to lots of little stuff, but also because we've increased the amount of heat, right? Remember, heat is just increasing the amount of random collisions that are going to take place. Right, so here's my cell, right? Here's some molecules in my cell. Here's the surrounding aqueous environment. What do these arrows represent?
Movement.
Yeah, it's kind of showing you the direction of movement that it has. So if our cell takes these individual molecules in green and sticks them together, right, one of the byproducts of that process is heat, right? So what happens to the surrounding environment?
It's not moving faster.
Yeah, right, they're absorbing that heat. We've got more random movement, so we increased or decreased the amount of entropy. We've increased the amount of entropy. All right. So just to give you some idea, if I eat 10 kilograms of food, right, I can make about 1 kilogram's worth of stuff. Right, I gotta break down a lot of material just to make a little bit. So I'm making a really big mess, even if I am cleaning a little bit of it up. in the end.
Professor, also anabolic release heat.
Anabolic?
Yes.
It can, yes. Right, so anytime I have a chemical reaction, well, depending on the type of chemical reaction, you can
So
have
this one
release
is
heat.
anabolic, that one is also.
Yes, this one is, yes. But in both cases, you're gonna have energy lost as heat, because we're transforming it in both cases. Right, so even if we're transforming it because we're breaking bonds down here, or because we're doing something else here, some of that energy is gonna be lost in the form of heat. Right, so remember, if we're constantly losing energy in the form of heat, that means we're running out of useful energy, so we gotta eat. Right, we have to have a continuous supply of this so that we can continually do that. So how do we talk about energy? We discuss it in terms of this thing called Gibbs free energy. It's named after the guy who sort of coined the concept. Right, so let's just say I'm starting out with, I don't know, molecule A and molecule B, and then it's going to become molecule C and D. Right? What's going to determine if this will take place spontaneously? Spontaneous means I don't have to add extra energy into my system here to make it happen. It'll take place. What makes this spontaneous? It's this thing Gibbs free energy. So it depends on how much energy the system already has. All right? So we measure Gibbs free energy usually in kilocal per mole or kilojoules per mole. I think this is the one you'll typically see. So what we're going to look at is the change in the amount of energy between our products and reactants, or delta G. Right, so I want to look at how much energy do I have in my products versus my reactants. So delta G is the energy of my products minus the energy of my reactants. So let's just brainstorm here. Right? What is going to be favorable? A positive delta G or a negative delta G? So think about what that means for a second. Talk with your neighbor. What would it mean if I had a positive delta G or a negative delta G? And which one of that would mean a spontaneous reaction, i.e. I don't have to put any energy in? So take a minute, talk about that, and we'll see what we come up with.
if the numbers, I know. If it's just higher, if the numbers higher than the the native will not energy. Because it will process.
All right, so somebody have any idea? What do you want? Negative delta G? Okay, how do I get a negative delta G?
If you're real.
So my products have to be less than the
Less.
reactants.
Yes.
So products have to be less than reactants. So what that suggests is that what you start with has more energy than what you end
Yeah.
with,
Mhm.
right? If I had a positive delta G, that means that my reactants would have more.
Energy.
Is that right? Which one did you wrong?
The product.
Yeah, you're right.
There we go. No, I do. That's why I flipped these around. Thank you. There we go.
Yeah.
Right, because what this means is that what I start with has less energy than what I ended up with. Well, that means I had to add some energy into the system then, right? So, a negative delta G is going to be one that's favorable where the products have less energy than the reactants. Right, so negative delta G is what's going to make things spontaneous. Okay, so look at these two graphs. Right now, we're going to use two different terms. A negative delta G is going to be exergonic, and a positive delta G is called endergonic. So you can hold up the number of fingers corresponding to A, B, C, or D when you get that far. Gives free energy. Okay, so which is larger, the G for your products or your reactants?
Yeah.
Okay, so your reactants, G of your reactants is greater than G of your products. Okay, so if I did what I'm supposed to do, which is products minus reactants, what should I get, a positive or negative?
Negative.
Negative delta G. What do we call this?
Exergon.
Okay, so let's look at the bottom one. Now,
Products are greater than
Reactants are
reactants.
less.
alright, so now your G of reactants is less than the G of your products. So, what's our delta G?
Positive.
Positive.
Positive, so we call this
And they're gone.
endergonic. Okay, so, what's our answer here?
C.
C.
Yeah.
Sometimes it's easier, so see what it's asking you and just go interpret the graph,
Yeah.
right? I mean, you could always go through each one by one, but so just recognize it's not the only strategy.
Variety.
All right. So, which of these two is spontaneous?
Top one.
All right, so another sort of an analogy to think of here. In an endergonic reaction, right, we have to input energy. It's kind of like when we're thinking about the concentration gradient. If I want to go from low to high, it takes energy, right? I'm trying to push a ball up the hill. This just doesn't happen. On the other hand, right, it'll roll down the hill just fine. I don't have to add any extra energy for this thing to go down the hill. Right, so our exergonic is spontaneous. Okay, so some terms that we're going to put together, right? Things that are a positive delta G are energetically unfavorable or non-spontaneous, which means that things are energetically favorable or spontaneous are our negative delta G. Right? Remember we said that this is exergonic, these are endergonic. And we mentioned a few minutes ago, right, if we're going to build things up, that requires an input of energy. So if it requires an input of energy, is that going to be spontaneous or non-spontaneous? Non-spontaneous. So building or anabolic reactions, positive delta G, right, input of energy. Catabolic reactions, where we're breaking things down, are generally exergonic, spontaneous, or energetically favorable. Right, so if you see any one of these terms, you should be thinking about the other ones at the same time. They all go hand in hand. Now, just because something is spontaneous says nothing about how quickly it will take place. Right? Remember that molecule of sucrose that I showed you a few minutes ago? If I had sucrose in a beaker, right, it's spontaneous. It's energetically favorable for sucrose to hydrolyze into glucose and fructose. But if I just left a beaker on the benchtop, I'd have to wait 200 years for that to start to happen, right? So just because something is energetically favorable says nothing about the time span for something to take place, right? Now, if I need glucose, and I've got a bunch of sucrose that I just ate because I just put a bunch of sugar on my cereal, can my cells wait 200 years for that sucrose to spontaneously break into glucose and fructose?
Probably not.
Right? You need that glucose really quick. So, what are we going to do to speed up that process?
Enzyme.
We're going to use enzymes, right? So we're going to come back to that in a few minutes, but that gives you some idea of why enzymes are important. Just because something is energetically favorable doesn't mean it's going to happen on any time scale that's reasonable. We still have to speed up these sorts of chemical reactions. Right now, you might remember from chemistry that, right, enthalpy, entropy, and temperature all play into this. You don't need to know these details, right? For our purposes, it can be really helpful, especially if you're headed towards biochem, but I'm not going to hold you guys to this. You just need to know positive and negative delta G's. Right, so Again, if you're biochem and you want some practice, work on this, but otherwise you can ignore this slide. Right? Just remember positive, negative delta G. Alright, so our synopsis here. Whether or not a reaction is going to happen is determined by whether or not we have a positive or negative delta G. Right, so catabolic, we're generally increasing the amount of entropy in a negative delta G. Right, oftentimes in a positive delta G, we're actually going to decrease entropy some amount. Right, oftentimes a positive delta G is associated with an increase in potential energy. Right, so for example, photosynthesis, we're going to take kinetic energy in the form of sunlight, right, photons, and convert that into chemical energy, so we're increasing the amount of potential energy in that case. So we're using kinetic energy and transforming it into potential. A negative delta G is usually a loss of potential energy, right? So for example, in cellular respiration, we're taking glucose, which has lots of potential energy in those bonds, and we're going to convert it, right, transform it, and in the process, we're going to have a net release of kinetic energy. All right, so what does it mean if delta G equals 0? Yeah, nothing's happening. All right. So this is just showing you the same sort of thing in a more diagram sort of format. Right, so for example, diffusion, right? We have more work capacity here, we have greater potential energy as diffusion takes place, then we're going to have less free energy, it's going to be more stable, we can do less work with this, right? We start with a molecule of glucose, there's more free energy here, right? We've got a lot of potential energy here, and as I break this down, right, we're going to release the energy to do work, for example, right? Our delta G is going to be negative for this sort of thing. So, living systems have a couple of challenges because we're bound by the first and second law of thermodynamics, right? The first thing is we have to make sure that reactions, even energetically favorable ones, take place in a reasonable time period. Right, so that sucrose hydrolyzing to glucose and fructose is favorable, but it still wouldn't just happen in a reasonable time period for my cell to survive. So, as we mentioned a minute ago, the solution to that are enzymes. We can speed up that chemical reaction. But we have another problem. There are lots of reactions that have to take place in the cell that are energetically unfavorable, right? They're endergonic reactions. My cell has to build these things, right? So, we need some way, right? If it's unfavorable, that means we have a positive delta G, which means we have to put energy in. Where are we getting that energy? Well, we're going to couple the energy that's released from favorable reactions to power reactions that are energetically unfavorable, right? So here, I'm releasing energy that I can use to power these sorts of reactions. So we refer to this as coupling. We can take energy that's released over here and use it to power reactions that need to take place over here. Can you think of a molecule that would be really good to sort of, I don't know, take energy from one area and then go use it over here? ATP, right? There are lots of different molecules that we use for energetic coupling. ATP is one of the most important, if not the most important, right? I'm going to use the energy that's released from favorable reactions to make ATP. Then I can take that ATP and use it to power reactions that require an input of energy. Okay, so let's look at enzymes. So if I take oxygen and hydrogen, I can get water. If I take a wooden log and I light it on fire, I'm going to end up with carbon dioxide and water. Take your notebook paper, it combusts, you end up with carbon dioxide and water. If you eat your Wheaties, same thing. It's going to end up ultimately as carbon dioxide and water, right? All of these have a negative delta G, right? So all of these are energetically favorable. But have you ever just seen your notebook paper spontaneously burst into flames in front of you?
Not today, at least.
Not today, at least. All right. Why not? Right? If this is energetically favorable, why don't I see this happening? What's that? Yeah, it takes some energy to get the process started. Right? It's still energetically favorable, but it takes some activation energy to get the chemical reaction to begin. Right? So even endergonic reactions require some activation energy. Yes. So even an energetically favorable reaction is going to require a certain amount of energy for the reaction to get started. All right, so we talked about this a few minutes ago, right? This is energetically favorable, but if we don't have a catalyst, we gotta wait a couple of hundred years. So let's add a catalyst, right? So how does a catalyst speed up the reaction, right? What's going to go on here? So here's a sort of representation of our reaction, right? We have the free energy on this axis, and then we're going from reactants to products. So we're going to start here, right? Our reactants are in a stable state. But in order for us to go from the stable state and go from reactants to products, we have to go through what we call a transition state. We have to transition from being nice and stable to being temporarily very unstable before we get transformed into product, right? And when things are unstable, that's a much higher energy level. So we have to go through a little bit higher energy level before we can become a product. Right? So once we get over this activation energy hump, right, it's all downhill. We can make products. All right, so here's the more formal diagram for this, right? We have free energy here, right? We're starting as reactants, we go to products, right? We call this the distance from the energy that our reactants have to the top of this little hill here. We call this the activation energy, right? Or E sub A for short, right? This is the stable reactant. This is what we call the transition state or the unstable intermediate, right? It's somewhere in between reactants and products, but it's unstable. So if it's unstable, it's at a higher energy state. So how are we going to get something over this energy hump, right? This is really the barrier. Even if it's a favorable reaction, right? That's the barrier for the reaction to taking place. So, where does the energy for reaching this state come from? Well, if you were just sitting around in a chemistry class and you needed a reaction to go faster, what would you do?
Increase the temperature.
You'd increase the temperature. What happens when we increase the temperature?
Molecules run around hitting each other more.
Yeah, the molecules are going to begin to collide more because they're moving around faster, so they're going to collide with each other more, and they're going to collide with more force, right? So you increase the number of collisions, you increase the amount of force, so you increase the probability that they're going to hit each other with just the right amount of energy at just the right angle for a chemical reaction to take place. So far, so good? Right, so if these are my two things and I need to make a bond between them, right, if I'm at a lower temperature, they're slowly colliding at random, right, I might have to wait a really long time for them to just, by happenstance, collide with just the right amount of energy at just the right angle to make a bond, right? I can make that faster if I just speed things up, right? And eventually, they hit it just the right angle with enough energy to make that happen. So, if you're a biological system like a cell, is increasing the temperature a feasible way to make sure all the reactions happen? No. I don't know about you, but I don't want to hold my hand over a Bunsen burner just so my hand stays alive. Right? Like, temperature is important, but that can't be the determining factor. Right? So, we are going to use enzymes instead. Right? Instead of colliding things harder to get them over this energy hump, we're going to be tricky. We're going to say, well, instead of requiring this much energy, what if I know a way to reduce the amount of energy that it takes? So that the amount of energy they already have at, say, body temperature is sufficient to get over this hump. Right? This is what a catalyst does. Right? This is what enzymes do. Enzymes are biological catalysts. They are going to reduce the energy that it takes to reach the transition state. This is still the transition state. It's still an unstable intermediate. But we don't take as much energy to get there. So somehow my enzyme is going to reduce the activation energy. That's the key. Right? So, hint, hint, wink, wink, nudge, nudge. Things you should be able to explain in your sleep. How is it that enzymes speed up a chemical reaction? They reduce the amount of activation energy required for a reaction to take place. All right, now look at delta G. Does the enzyme change the delta G for a reaction? No. Right? Delta G is just the difference between the energy in the products and reactants. Right? Whether or not it's catalyzed or uncatalyzed, my reactants have the same amount of energy and my products have the same amount of energy. So the delta G does not change. Right? Another way to think of this is a good analogy. Right? Here's your reactants, right? And they've got some kinetic movement, right? Some kinetic energy. They're bouncing around. Right? But the energy that they have is typically not sufficient for them to get over this activation energy hump. Right? Now, maybe every once in a while you get like one that gets a little extra energy and whoop, okay, maybe it makes it over. Right? But for the most part, virtually everything that's in your system doesn't have the energy to get over that barrier. Right? So what does an enzyme do? Well, it reduces that barrier so that now the energy that they already have is sufficient for them to go to product. Let's see if this thing will play. Okay, so this is just a simulation. This is 1.8 microseconds. Supercomputers simulated interactions between molecules. The reason why I show you this is because, again, even in most of the animations that I give you, the cell looks like this big, giant, empty space, right? And every once in a while you see a protein like floating by. It's a really crowded environment, right? We're not showing you all the water molecules that make up the environment that these guys are packed inside, right? So collisions are happening between things all the time. Okay. So, enzymes. Any questions about enzymes so far as what they do? We'll talk about how they do it here in a second. Okay, so we want to make a reaction take place on a reasonable time scale. So, there are different types of biological catalysts, right? The most familiar one, the one we're going to talk about the most, are enzymes. Enzymes are protein catalysts. There are, however, some RNA molecules that can catalyze chemical reactions as well, and we call these ribozymes. In fact, one of the most important ribozymes that exists is in the ribosome. In the ribosome was made up of protein and RNA. And the RNA in the ribosome is actually responsible for making the bond between two amino acids. What do we call that bond? Peptide bond, right? So actually building a protein is done by the RNA part of a ribosome. So the vast majority of enzymatic reactions in a cell take place via proteins, protein catalyst, enzymes, but there are some important RNA molecules that can do this, right? And you'll know that you're talking about a catalyst, a protein catalyst, if you see the term ending in ase, right? So sucrose can be hydrolyzed by sucrase. Ribozymes are RNA catalysts, which are fairly rare. So what we want to know is how do these things lower activation energy? Well, the first thing we want to understand is that enzymes are specific. What makes an enzyme specific for a particular substrate? You guys all answered this earlier already? Shape and chemistry. Right? This is this concept of complementarity. Molecular complementarity. So, here's molecule A. Molecule A is floating around and it bumps into B, C, and D. All right, now, there are some weak interactions that can take place when they bump into each other, right? But they're not sufficient for a substantial interaction between them, right? It's more of just a collision and a separation. But if you look at A and D, you can tell, right? Their shape is extremely complementary to each other versus these two. And in addition, there are a significant number of interactions that take place between these two molecules, right? So what kind of interactions could these be? Hydrogen bonds, what else? It could end up being a covalent bond. These are typically temporary things, so they're not often is it a covalent bond, but it could be. Could be ionic, could be hydrogen, could be van der Waals, hydrophobic, right? All of those will mediate the interaction between two molecules, right? So these guys are going to bounce, they collide, they go on their merry way. These two collide, and now they are associated with each other, right? They have a lasting interaction. All right, so these two are complementary to each other in that sense. Okay, good. It is here. I'll turn my on this slide. Right, this is molecular complementarity. I'll ask you about this. Okay, so here's a generic blob diagram of an enzyme. Right, here's a substrate. Right, this is where the two interact with each other. So what do we call that region? Active site, right? It sounds familiar from last week when we did enzymes, right? So enzymes are going to lower the activation energy, first of all, by binding the reactants, i.e. the substrate, in an orientation and in a conformation that's favorable. What do I mean when I say conformation? What does conformation mean? Fancy way of saying shape, right? So, right, remember I had my two reactants and they're just like bumping into each other, and like, you know, if I heat them up, maybe they'll eventually collide with just enough energy to make a product, right? Wouldn't it be a lot easier if I was an enzyme and I just went grab, grab? That's what your enzyme's doing, right? It's got the right shape to bind to the substrates, and it's going to put them in the right orientation, right? I'm not doing this, I'm not doing this. I've got them oriented in the right way to come together. Right? Now, we're not always building things, sometimes we're breaking them, right? So imagine if this was going to break, right? You guys have ever flexed a pencil? Did you guys ever do that back in the day? Twist, twist, and then eventually it just snaps? Right? It's a similar idea when we're breaking covalent bonds. Right? A bond is going to be stressed. So if I was going to be the enzyme here, I'd be stressing this bond, right? It's not broken, right? But it's stressed. This is the transition state. It takes energy for this to happen, right? but I've lowered the amount of energy that it takes because when I grab onto this, I flex, right? The active site flexes along with my substrate. They flex together. It's what we call an induced fit. It's kind of like when you put on a glove and you kind of stretch it with your hand, right? You've induced a better fit. Same idea. Your enzyme and your substrate are going to flex together to produce the best fit. And that's going to promote the transition state so that it takes less energy to get there. So we're going to put things together in the active site. The active site shape is going to put things in the right orientation and conformation. In addition, both our substrate and our enzyme are going to flex when they come together. This is this induced fit that promotes the transition state. Right? And so as a consequence, we lower the amount of energy necessary for this reaction to take place. That's the magic behind an enzyme. Putting things together in the right orientation and conformation and promoting the transition state. Right? So that interaction puts it at the top of the energy hump. But that energy hump is now lower than it had to be. Okay, so questions about how enzymes reduce activation. Yeah.
I don't understand how the induced fit promotes the transition.
So, on the enzyme, when I grab my substrate, we both flex. This is the consequence of me interacting with the substrate. This is the transition state. So normally, for this to get to the transition state, what would it have to do? It would have to collide hard enough, right, to stress that bond in just the right way. So what do? I don't know, I heat up the reaction, right? And if it hits at just the right angle, we stress this enough, right? But that energy hump is higher than if it just comes and hits me at room temperature and we interact just right to stress that bond right.
So the reaction would be it breaking?
The reaction is when it finally breaks,
Okay.
right? But just hitting the enzyme and interacting with the active site gets us to the transition state.
Alright.
All right, so here's an example of this. So this is just showing you the wire diagram for an enzyme called hexokinase, right? And this is hard to see in this, so I'm going to show you this animation. Right, so here's the enzyme hexokinase. In fact, this is one of the models I passed around earlier. It's going to use glucose and ATP together, right? And what happens? It goes from like this to like this. It actually flexes around. So I'll pass these around in pairs. So make sure you keep this as a pair because you want to compare them side by side. But you can kind of see this, right? Here's the open conformation with the active site, right? Ready to receive the substrates. This is the shape once the substrates are bound, right? Can you see the difference? Right? It's kind of like a hinge. It's closed a little bit. It's fitting around the substrate. Right? So this is one example of an induced fit. Right? And then in the active site, it's going to catalyze the reaction. So in this case, we took a phosphate off ATP and stuck it on glucose. And then we release the substrate, or the products now. Right? So it's a temporary interaction. We catalyze the reaction. The products are released. Right, so there it is. Here comes the substrates. They fit in the active site. They flex together. Now the reaction is catalyzed. We have two different products. Those products are now released. So let's look at the active site. Right, here's a protein, and this is just showing you the backbone, and then the little popsicle colored sticks here represent different side chains from amino acids, right? So different color, different side chains. So they're going to have different properties. So if I fold this thing up, right, here's the active site, or the binding site, where it's going to interact with something. Right, you'll notice these amino acids are all along the entire chain. But when they fold up, they're all in this three-dimensional region of the active site. So what's going on, right? So now we're going to take this and we're going to represent it sort of this way, so we just see the shape. Right? So here's my ligand. What do we say a ligand is? Anything that is bound by something else. So this protein is going to bind to this ligand. So this guy, you'll notice what this shape is what compared to this?
Complementary.
Complementary. Okay, so these two can interact with each other, right? They have the right shape, so this has the right shape to fit into the binding site, aka the active site. And then we have all these non-covalent interactions. They're going to mediate the interaction between these two. Right? So what are some of those things? What kind of non-covalent bonds can be taking place? Well, here's my ligand in orange, right? And gray is the backbone of my protein, and then right, the yellow here are the side chains for serine or threonine, right? That's like this guy or this guy. This is glutamic acid side chain in blue, so that's this little blue popsicle stick. Right, so now it's showing you the close-up chemical structure of these amino acids and of the substrate. So what's happening? I have an OH here from serine, and over here I have a P double bond O here. So, right, this should have a partial,
Negative.
and this will have a partial
Positive.
positive. So this oxygen is happy to make a
Hydrogen bond.
hydrogen bond with this guy. So there's one temporary interaction between my amino acids from my protein and my substrate, right? Here I have an arginine that has a positive charge, and here's an oxygen with a negative charge. So this is an example of an ionic bond. Okay, and then what do I have over here? I have more hydrogen bonds, right? I have an OH group hanging off of my substrate, right? And I actually have that oxygen making a hydrogen bond with this hydrogen here, and then this C double bond O is making a hydrogen bond with this hydrogen here. Got another hydrogen bond over here, another one here, and another one here. Right, so these are all non-covalent bonds, but that's a lot of stuff happening, right? That's going to allow this substrate to be held in this pocket, at least temporarily. Okay, so it's both the shape and the chemistry that determines the molecular complementarity. Spend some time with this diagram. And Tim, wait. Fetch back. Oh, let's see this one. Is this one good? This one you gotta download. Oh, okay. Alright, so here's our protein. This is maltose binding protein. Here's a maltose, triose. So here's the three things. So what are all these little red lines representing?
Bonds.
Yeah, those are hydrogen bonds. Right, so right, if we zoom in here, right, these are all the side chains of the active site that are highlighted in blue. Right, our substrate is this yellow thing in the middle. So those represent the hydrogen bonds in between the amino acid side chains and the substrate. Right, and then if we looked at the surface, instead of using a wire diagram, if we looked at the surface, right, there's the three-dimensional surface. Right, so it's fitting into this pocket, quite literally, the same way, remember I showed you this guy, the little blue thing in the middle, right? It's got just the right shape to fit into that pocket, but it also has the right chemistry to make those sorts of temporary interactions. Yeah.
Are these supposed to like fit, or is it just showing?
It's just showing you without substrate bound, with substrate
Right.
bound.
So it's like one is like more condensed, kind of.
Yeah,
or
because it's
flex.
gone from like open
Right.
to slightly closed. Okay. In the active site, right, the environment here is not necessarily identical to the rest of the aqueous environment in the cell. So, right, you might have a low pH environment in the active site that could alter the chemistry of that site. In addition, the R groups sometimes directly take part in the chemical reaction. So they might actually temporarily make a covalent bond between things, right, during this chemical reaction, and then they go back to being the same way they were when we started, right? Because what are the key properties of a catalyst? They can be used over and over again, right? So even if it takes part in a covalent bond, it's a temporary thing, right? It might be breaking this one, making one temporarily so it can build another covalent bond over here, but when it's done, it's back to the exact same thing it was when it started. Okay, so here's our synopsis, right? The active site's going to lower the activation barrier by orienting things correctly, straining any bonds that we have, making sure that it's a favorable environment, right? And it might take part in some covalent bonds temporarily. Okay, so here's another example of this, right? I have my enzyme. I have two substrates in this case that are going to fit into the active site. So we've got weak interactions here that are temporarily going to hold this together. So this is an enzyme substrate complex, right? We lower the activation energy, so we catalyze some sort of chemical reaction here, and now we're going to release the products, and now I have an active site that's available to do this all over again, right? And we can just keep doing this. My enzyme is unchanged by this process. Okay, any other questions about activation energy or enzyme shape, active site role? All right, this is just another view of the same thing, except now we're looking at our sucrose and sucrase, right? So here's the enzyme, here's the active site for sucrose to fit. So our sucrose binds, so I have an enzyme substrate complex. We're going to hydrolyze the chemical reaction to hydrolyze the bond here, or catalyze it. Now we have our products, right? And we can do this tens of thousands of times per second as opposed to waiting a couple hundred years for this to just happen on its own. Enzymes oftentimes don't function alone. They need some helpers. So sometimes they use what we call a cofactor. So cofactors are usually things like a metal ion, right? So it's got to use a metal ion to do its job. So for example, hemoglobin requires iron in order to grab oxygen, right? So it's got a cofactor, right? DNA polymerase, the enzyme that's going to build DNA for us, requires, for example, magnesium as a cofactor to function properly. We also have things called coenzymes. These are usually small molecules, oftentimes that are organic biological molecules, that are required for an enzyme to complete its function, right? So we're going to see ATP, FAD, NAD, coenzyme A, all of these we're going to see over and over again later throughout the course, right? Some of these are really important for energy coupling, right? So we mentioned ATP earlier. And then these are usually temporarily associated with an enzyme, right? They hang out with the enzyme temporarily and then they leave and go somewhere else to do something else. Prosthetic groups are pretty much a permanent part of the enzyme, right? They're not made out of amino acids. They're a small molecule that the enzyme has to use, and it's always stuck with it. So heme is an example of this in hemoglobin, right? So here's a molecule of hemoglobin. There's 4 different subunits that are stuck here together. Heme is this group in the middle. It looks like this, right? So this molecule is being held by hemoglobin, the protein, right? And then that is actually holding on to an iron atom. So it's both got the cofactor and a prosthetic group. And it uses this to grab onto oxygen, right? If our hemoglobin didn't have iron or didn't have heme or both, it couldn't hold on to oxygen, right? So it needs these things to accomplish its job, right? So prosthetic groups are pretty much permanently attached to the protein. Coenzymes associate temporarily during a reaction, right? Metal ions can either be, permanently a part of the enzyme or transient. Jasmine, you had a question?
Yeah, so for the ones that have metal
the cofactor is the metal ion. All right. Think about your enzyme reactions. What happened when you ran your time course? If you just lined up all your tubes, right? You had like 0 minutes, 5 minutes, 15, 20, 25. What was the visual difference between them by eye?
More time with yellow.
The more time, the more
Yellow.
right? Yellow was the product. So the longer the enzyme did its job, the more product it made, right? If we had graphed our data in a slightly different version of that experiment, right, we could have followed how much product is being made over time, right? Or we could follow how much substrate we have over a given amount of time, right? So what we can plot here is, for example, the reaction rate versus the concentration of substrate. So let's think about two things. We're going to think about two different plots. So one is what you did, right? Where you have time, and then you're basically looking at the amount of product, right? And it probably looks something like this, right? So over time, I got more and more product, right? But we could also think about how much are we making per unit of time? Like, what's the rate of the reaction? So on this axis, we're saying how much do we make per time? On this reaction, we're saying it depends on how much substrate I have. So let's say that we ran a 5-minute reaction, right? And we ran like a dozen of them,
Oh.
right? I ran a 5-minute reaction with how much substrate?
0.
Like none, right? And then I ran a 5-minute reaction with, I don't know, whatever this is, 5 millimolar substrate. And then 10 millimolar substrate. I ran another 5-minute reaction, right? And so I could measure how much product do I make in that given amount of time, depending on how much substrate I have. So what do you notice is the trend here, right? What's the distinction between this line and this line?
Substrate increases linearly. The other one not so much.
So if we don't have an enzyme, yeah, right? It's like, okay, yeah, over time we're gonna make more and more stuff. But the rate, i.e. that slope, is very low. But if we add the enzyme, what do you notice?
Boom.
Boom, right out of the bat, we're gonna be creating a lot of product very, very quickly. And then at a certain point, we're not going any faster. We're still making product, but we're not making it any faster. Why not? Why would I not be making it any faster at a given point?
Carrying capacity.
Yeah, right? So imagine that you have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, I've got 10 enzymes in my reaction. Okay, and I put just a little bit of substrate in there. How fast is my reaction going to go? Yeah, I'm going to get one product like instantly. Okay, but what if I double the amount of substrate that I put in? I'm going to get twice as much reacted in the same amount of time. So what if I added some more substrate?
Oh, you're limited by your number.
3 times, right? So when am I going to plateau? Yeah, when all of my enzymes are going to be reacting with substrate. So when my substrate concentration is high enough that all the enzymes are constantly working. Right? So the rate of reaction doesn't just depend on the things that we've talked about so far. It also depends on the concentration of your enzymes in your substrate. Okay, how many of you guys have done the worksheet already? That's due next Thursday. Like one person? I know at least one person. Okay, so think about this, right? Let's say that you have an enzyme called pepsin. This pepsin is an enzyme that works in your stomach, right? And you want to know how is the reaction rate going to change at different pHs, right? So talk with your neighbors, sketch a graph of what you think this will look like. If you've already looked at our data from class, you might already have seen this. So make a prediction.
Yeah, we don't want that. We don't want that.
Just draw the Morgan graph that you got for beta gal. Beta gal? For the pH. It doesn't have to do like perfect numbers, we just want to see the shape of the curve. Got silent, so I guess we got a prediction. Just draw a line through it. Okay, so this was somebody's data for beta galactosidase. Right, so graphed, that's a really rough graph, but you get the idea. All right, now let's think about what we would expect to see here. What do you think the line should look like with this one?
Should hurt to the left a lot.
Okay, so where do you think the highest reaction rate or the most activity is going to be?
Around 4 maybe?
Around 1.5 or so.
Somewhere over here?
Yeah.
So maybe something like this? No?
No.
Okay, why not?
Or your maximum point would be around.
It'll go down.
Okay, so it should go down?
Shouldn't it be high since stomach acid is very acidic?
Yeah. So
Yes.
stomach
Okay,
is
so it should be high here and then what?
Should just
Low?
That's fine.
Yeah.
Now, like roller coaster, like you go up and then you go down.
Oh, so you think it's gonna go up? And down?
Well,
That?
not well,
Not like the lighter.
Okay. Well, I mean, what's shared in common between both of these?
It goes down.
It's decreasing.
It's high over here and low over here. Why? Okay, so that seems like a reasonable prediction. But then I look at this graph, and this graph is completely different. Where's the best activity for this one?
Seven.
Seven.
Is this wrong, or is that wrong?
It's different
No.
Trick question. All right, they're different enzymes. Why did you think that this one was going to work better at a lower pH?
Because of acid.
Because it's in the stomach, right? Stomach acid is around a pH of 2. So you would imagine for a protein to function in that environment, right, it's got to work well. That doesn't mean it's going to work well at other pHs, right? A dramatic shift in pH could denature your enzyme, right? So for beta-gal, it worked really great around pH 7. But if we start to shift too acidic or too basic, right, that protein starts to denature. All right, so here's an example, right? So this is pepsin, for example. Here's chymotrypsin, arginase, right? Some of them work better even at basic pHs than they do at other pHs. So each enzyme, it's going to be, dependent on the enzyme. You can't just say all enzymes work great at neutral and they work terrible in acids or basic. You've got to know, you've got to test each enzyme. Okay, so what about temperature? Well, here's a typical human. 37 degrees is the optimal, right? So you would expect that most enzymes function best at around body temperature. Okay, now, there's some bacteria that like to live in like hot springs, really warm environments, and you would expect, right, that their optimal is going to be at a higher temperature, right? In fact, one of the enzymes that is the reason that molecular biology is where it is today is because we discovered that this enzyme is capable of making DNA in an organism that lives in these really hot temperatures. It doesn't denature even though the temperature is very hot. Right? That enzyme is called DNA polymerase, but it's from Thermus aquaticus. So we call it TAC. Right? So TAC polymerase allows us to do something called PCR. How many of you guys have heard of PCR? How many of you guys have ever watched any crime scene show ever where they do some sort of DNA forensics?
I think so.
They're going to use TAC, right? You guys are going to use TAC to do PCR in one of our labs later on. But as a part of that process, you have to heat the DNA up to very hot temperatures. And if we did that with a normal human enzyme, it's not going to function anymore. Right? So different organisms, right, have different environments where they live, so they're better adapted. So you might expect the proteins for an organism that lives in warmer temperatures to survive better here. Okay, you guys went through these slides in the afternoon section as well, right? So the idea of enzyme inhibitors, whether they're irreversible or reversible, non-competitive or competitive, you're familiar with these? Okay, so I won't rehash those. The last thing I'll leave you with is this notion that molecules that bind to proteins can impact it, right? A protein might have more than one binding site. But the binding of a molecule to a protein can shift its shape. We saw that here, right, with a non-competitive inhibitor. All right, our protein has this shape, this guy binds, it changes its shape. Right? This is called allosteric regulation. So do we have that on the next slide up here? There we go. Right? The binding of a ligand to a protein can change the shape of that protein. Now, it could make that protein more active, it could make it less active, right? It just depends on the protein and the ligand. So we can upregulate a protein activity, we can downregulate a protein's activity. This is called allosteric regulation, right? And this is reversible. This is a temporary interaction between, right, a small molecule or another protein or something else, right? It doesn't have to be a protein-protein interaction. It can be a protein and a small molecule or something else. Right, in the same way, right, so in this case we have a protein that's inactive, we have a protein that binds it, we change the shape, and now it's active, right? So in this case, this is a positive allosteric regulation. We can also allosterically regulate a protein by putting, like for example, a permanent modification, like a phosphate. When I say permanent, I mean something that's covalently bonded. Right, so this guy's inactive, we add a phosphate group, the protein shape changes, and now it's active. Right? And so it does its job. What would happen if we cleaved off that phosphate? It'd go back to being inactive. Right? So we can regulate, we can make a protein, but just because I have the protein doesn't mean it's going to do anything. Right? I can make the protein and keep it off until I need it on, and then flip the switch, so to speak, to have that protein active. So we can allosterically regulate these guys. Okay, so that's where we're gonna leave it. When we come back, we will deal with energy coupling on Tuesday. Awesome.